Today we are going to be discussing about a gram staining. So this gram staining is one of the most commonly used stain in the microbiology lab, and it was developed by Danish physician Hans Christian Gram in 1884. And gram staining is still the cornerstone of bacterial identification and taxonomic division. So this gram stain is a differential staining procedure which separates most bacteria in two groups, that is on gram positive bacteria and the gram negative bacteria, where the gram positive bacteria have a thick layer of peptidoglycan and then the gram negative bacteria have a thin layer of the peptidoglycan. Now coming to the principle, since a gram staining is a common technique used to differentiate two large groups of bacteria based on the different cell wall constituent, the gram stain procedure distinguishes between gram positive and gram negative by coloring the cell red or violet. So in this gram staining, they involve it involves like four steps. Four steps or four reagents, where you are going to be using the four reagents. So the first reagent you use is the application of the primary stain, which is crystal violet, which stains the cells purple or blue in color. Then you use the application of a mordant, which is gram iodine solution, which is added to form a crystal violet iodine complex, and all cells continue to appear blue in color. The next reagent used is a decolorizer, which is an alcohol or acetone. This decolorizer distinguish gram positive and gram negative cells, and the organic solvent such as the acetone or the alcohol extracts the blue dye complex from the lipid rich, then thin wall gram negative bacteria to a greater degree than from the lipid pore. So thick wall gram positive bacteria, the gram negative bacteria appear colorless and gram negative bacteria remain blue in color. The next is the application of the counter stain, that is safranin. The red dye safranin stains the decolorizer gram negative cell red or pink in color. So the gram positive bacteria remain blue in color. Let us see the requirement. What are the requirements we need before we proceed to the procedure of gram stain? So you need a glass slide, you need a sterile inoculating loop, you need a Bunsen burner, you need a crystal violet, gram iodine, alcohol, safranin, microscope, and of course the specimen. So before going to the procedure, for the specimen you can take directly from the culture with the growth, or from the sample directly, or from the broad culture with growth. And remember before performing the staining, you have to make this smear should be very good. It should not be too thick, it should not be too thin. So let us see the procedure now. Firstly, you have to flood air dry heat fixed smear of the cell for 1 minute with crystal violet staining reagent. Then remember, the quality of your smear should be good. It should not be too thick, it should not be too thin. Then you wash the slide in a gentle and indirect stream of tap water for 2 seconds. Then again flood the slide with the mordant, that is gram iodine. Then wait for 1 minute. Wash your slide again in a tap water for 2 seconds. Then now you have to flood with decolorizing agent, that is acetone or alcohol. It depends on you which one is available in the lab. Then you have to wait for 15 seconds, or else you can just do one thing, you can drop by drop to slide until the decolorizing agent runs off from the slide, runs clear. So the next thing is you have to flood the smear with a counter stain, that is safranin, and wait for 30 minutes to 1 minute. Now the last step, you have to wash your slide in a running tap water, and make sure that no color appears in the effluent, and then blot your slide with an absorbent paper or a tissue paper. Now you have to observe the result of the staining procedure under the oil immersion, that is 100x in the microscope. So let us see the interpretation. As I have said in the principle also, that the gram negative bacteria will stain pink or red in color as they have a thin peptidoglycan, therefore they retain the counter stain red or pink in color. That is why they happen to that is why they are stained pink or red in color. Here I have given the image itself, the first one, which is pink in color, you can see clearly. And then for the gram positive bacteria, it will stain purple or blue because it have a thick layer of peptidoglycan. That is why they retain the crystal violet color, that is purple or blue in color. Here you can see in the given image. Now how do we do the reporting of the gram stain, or how should it be reported, is that the report should include the number of bacteria present, whether many, moderate, few or scanty, then the gram reaction of the bacteria, whether gram positive or gram negative, then the morphology of the bacteria, whether it is cocci, diplococci, streptococci, rods or coccobacilli. I hope you all remember this morphology of the bacteria. Then the presence or the number of pus cells, because sometimes the pus cells might be present in the gram stain also, so you have to report that also. Then the presence of the yeast cells and the epithelial cells. Now you have to, for the quality control, you have to always check the new batches of the stain and reagents for correct staining reaction using a smear containing non-gram positive and gram negative organisms. So some of the suggested quality control for the gram positive is Staphylococcus, 80 ccs. Then for gram negative we have Escherichia coli, 80 ccs. So these are some of the suggested quality control organisms. And also there might be some like common errors in the gram stain, like which include like the wrong labeling or too thin or too thick smear, I have told you earlier. So then inadequate fixing, then the stain deposits, then over decolorization, or like if you have not followed the time given, where for example in the decolorizer, where you have to use only for 10 to 15 seconds, you might get confused and use for 1 to 2 minutes. So these are the common errors to be made, so you should be very careful while performing the stain. So this gram stain is a differential stain which distinguish the bacteria into gram positive bacteria and gram negative bacteria. So one example of gram positive bacteria is Staphylococcus aureus, and one example of gram negative bacteria is Escherichia coli. So this is all about the gram staining.